Thank you, Twanelo. Yes, my name is Nobuhle Majosi. I'll be chairing this session this morning, the health and biomedical. So first things first, the presenter has 20 minutes to present, but then we request that you do 15 minutes, and then the last 5 minutes will be for questions and answers. Okay, our first presenter is Lerato Mabe. She is a post-doctoral researcher with the Advanced Food and Agriculture in the CSIR. Her presentation is high sensitivity nested PCR for enhanced diagnosis of bovine brucellosis. Welcome.
Down for next. The next slide. Okay. Thank you. Thanks. I definitely need that one. Better? Good morning, everyone. I am Lerato Mabe. I feel like I need to sing just to shake the nerves off a little bit, okay? As Dr. Majosi has said, I am a post-doctoral research fellow in the aquaculture and animal health group within the Advanced Agri and Food Cluster. Today I'm just going to talk about some of the work that we've done developing a high sensitivity nested PCR to enhance the diagnosis of bovine brucellosis. This work formed part of my PhD, which I did under the supervision of Dr. Isa Suleiman and Prof. Oriel Tekiso. Bovine brucellosis is an important but often underestimated disease in South Africa, and our aim was to develop a diagnostic tool that is more reliable, that is faster, and that is safer to use than what's currently available. Okay. So bovine brucellosis is a bacterial disease that is caused by Brucella abortus, particularly in cattle. It significantly impacts both the health of animals as well as the productivity of small-scale and large-scale farming operations in South Africa and in many other countries. It is also a zoonotic disease. This means that it can be transmitted from animals, or cattle in this case, to humans. And this poses a significant risk to farmers who work directly with these animals, veterinary doctors, laboratory technicians, as well as anyone who comes into contact with infected animals or works with infected animal carcasses. With a global incidence of approximately 500,000 cases per year, unfortunately in South Africa, the incidence of human brucellosis is currently unknown because there's currently no active human brucellosis surveillance program within the country. And because this pathogen can infect multiple organs or any organ system in the body, clinically diagnosing it is very difficult because it produces symptoms that are similar to many other diseases. So this is a serious problem when it comes to human health, because if left untreated, the disease can cause chronic debilitating illness that has long-lasting effects. Unfortunately, available diagnostic techniques have a lot of challenges. For instance, bacterial culture, which is currently the gold standard test for diagnosing brucellosis, is very time consuming. It takes a long time, approximately or it takes about weeks to months for the diagnosis to be complete. And also, it exposes laboratory personnel to dangerous pathogens as they have to work with live bacteria. While serological tests are considered relatively safer and faster than culture, they lack sensitivity, which means that positive cases of brucellosis can be missed while using serological tests. But also, they can cross-react with pathogens that produce similar antigens as brucella, so thereby leading to false positives. And that is also an outcome that we do not desire. In South Africa, the diagnosis of bovine brucellosis currently is based on a combination of these two methods. But because these methods are either slow or lack sensitivity, it means that outbreaks often go undetected, allowing the disease to spread uncontrolled and posing or leading to significant economic losses in the livestock sector, as well as posing ongoing health risks to humanity. So this brings us to the heart of our work, where we then turned to molecular diagnostics in order to mitigate some of these challenges. And our aim was to improve the time it takes to get a brucellosis diagnosis, as well as to improve the reliability of the assays. So what we did was to develop a nested PCR assay and test this nested PCR assay directly and compare directly with an existing PCR protocol. Our work included obtaining milk and serum samples from cattle across different provinces in South Africa and rigorously testing both methods side by side. This gave us a clearer picture of how our assay performs in the real world. Okay, so here what you see is just a side-by-side comparison of the performance differences between the published PCR protocol, which I will refer to as the reference PCR from now on, and our nested PCR. This is just one of many examples, but I couldn't really show everything for this presentation. So what you are seeing here, shown by the arrows, is, can I point with this thing? Okay. So what you are seeing on this side, right, is this right, my right, left? Yeah. Yeah. So what you're seeing on this side is the performance of our nested PCR, and on this side is the reference PCR. So what you can see is that the nested PCR was able to detect Brucella DNA even where the reference PCR failed. This is just another example showing you our nested PCR picking up Brucella DNA where the reference PCR couldn't. So this just shows you how efficient the nested PCR was against the reference PCR. And what we also noticed was that the nested PCR was much more, it was not just sensitive in picking up infections, but it was much more robust than the reference PCR, as it was able to resist inhibitors of PCR, as well as it showed resilience to low DNA yield and low DNA purity. And that's the reason why with the nested PCR, we didn't get a lot of the non-specific banding patterns that you see with the reference PCR. When we compared the sensitivities of the assays, the reference PCR had a very low diagnostic sensitivity of about 3%, whereas the nested PCR boasted a 100% sensitivity. And when we compared the performance of the nested PCR in milk and in serum samples, we found that the assay performed consistently across sample types, highlighting it performed consistently across sample types, highlighting its ability to perform across various sample types. So what you can see here is that the nested PCR actually picked up more brucella in both milk and serum samples, whereas the reference PCR failed to pick up some of those infections. And this just highlights the value of the nested PCR in confirming suspected cases of brucellosis. Okay, so while this was exciting news for us to see our assay performing so brilliantly, it also was a cause for concern because a lot of the samples that we had received and tested with our nested PCR had been reported to have tested negative for brucellosis by our sample donors. So this just suggests that the incidence of brucellosis in South African cattle could very well be higher than anticipated. And this just highlights the existent diagnostic gap that critically needs to be addressed, which is the reason why we decided to develop this nested PCR. Okay, so while that was exciting to see these good results, before we got ahead of ourselves, we decided let's just make sure that what our assay is picking up is actually Brucella abortus. So we took some of the nested PCR products that came back positive for brucellosis, and we sequenced them, and then we blasted them against an entire sequence database on GenBank. Now, this sequence database will have the sequences and genomes of anything you can think of that's there on Earth, human beings, microorganisms, macroorganisms, everything that you can think of. So when we blasted our sequences against the database, we found, and this was the raw sequences, we found a 97% nucleotide match exclusive to Brucella abortus sequences in the database. And then what we then did was to run a phylogenetic analysis, and that analysis showed us that our sequences, which are depicted by the letters and numbers, they clustered with known strains of Brucella abortus. This gave us a strong confirmation that what we were picking up was actually Brucella abortus, and that our assay is actually accurate and specific. Okay. So having done that, we then decided to continue and validate the assay on all the samples that we had received, totaling 441, which included milk and serum samples. And then from the results, we can successfully say that we have developed successfully and validated a nested PCR that can detect Brucella abortus, specifically Brucella abortus. And we are also happy to report that this assay has now reached a technology readiness level 6. A technology readiness level 6, which means that the assay is now ready for deployment or further scale up. And we are also excited to announce that this technology has been accepted at TRL level 6 as a technology demonstrator by the CSIR TD panel. So in conclusion, the work that we've done goes beyond just developing a more sensitive diagnostic assay. by developing an assay or an assay that is improved in sensitivity, we are not just advancing science. We are protecting livestock, we are securing livelihoods, and we are safeguarding human health across South Africa. And I just want to leave it there for now. Thank you.
Thank you, Lerato, for that very, very interesting and critical presentation and research. Now we're going to take questions.
Hi, Lerato, and thank you for the very interesting talk. At the beginning of your talk, you indicated the lack of the surveillance systems in South Africa. That's what motivated you to go towards the nested PCR method. So my question is, now going beyond maybe South Africa, maybe looking in Africa or even worldwide, what was available and couldn't we use that and then calibrate it to our unique maybe conditions? Yeah.
Okay, thank you for the question. We did look at what was available in other parts of the world, and what we found was that many of the assays that are currently available, that have been developed for Brucella abortus, are not actually specific to Brucella abortus, because many of the sequencing technologies that came out a bit later on in the years. And because of that, Brucella sequences were not readily available to many scientists. So the assays that were developed prior to the coming out of the whole genome sequences of Brucella were developed thinking that these assays would be specific to brucellosis. But with the advent of whole genome sequencing and people having access to entire genomes, we were able to see that actually the assays that are currently available are not specific. So they can pick up brucellosis, but they can also, caused by Brucella abortus, but they can also pick up Brucella melitensis, Brucella ovis, Brucella suis. So we then developed a nested PCR that was specific.
So is this Brucella strain a South African issue, or is it shared by other countries, or is it specific to us?
Brucella abortus affects cattle in South Africa, but it does also affect cattle in other parts of the world. Yes, thanks.
Okay, next question.
Hi, thank you for the great presentation. You mentioned that traditional methods of detecting Brucella could take weeks up to a month sometimes. So with your current pipeline, how long is the detection time? And also, are there any strategies that maybe you've developed for on-field or within the farm detection of this pathogen? Thanks.
Thank you. Okay. With collecting a field sample, extracting DNA, and testing that extracted DNA with our nested PCR, it could take a day to get a result. But that would depend on how long it takes also to get a sample from the field into the lab. DNA extraction takes maybe an hour, 30 minutes. The nested PCR takes about 2 hours. So within a day, if you can get your samples into the lab, you could have your result. In terms of whether we've looked into, I'm not sure if you meant we looked specifically us. Okay, so we did develop another technology. It's called LAMP, loop mediated isothermal amplification. And I don't want to talk too much about that IP. I don't want to get into trouble, but we did develop that technology and we are trying to get it to be adaptable to the field. Yes, thanks.
Thank you for those interesting responses. Another question? We're left with 2 minutes of questions. Prof?
Thanks, Lerato. It was a good presentation, but I'm still not, having listened to you, which is a good presentation, I still don't understand what is NPCR. Does PCR stand for something?
Oh, thanks, Prof. So NPCR stands for nested PCR. So PCR stands for polymerase chain reaction. I'm not sure if you are familiar with polymerase chain reaction, but this is basically like a method of amplifying DNA or multiplying copies of DNA to make more of it so that you can be able to detect whatever pathogen or whatever you're trying to find within a sample, a small sample. Yes. So the nested PCR is just a derivative of the PCR, which works, so PCR works using a set of primers, and then the nested PCR works using 2 sets of primers. So where PCR will, the primers of the PCR will attach to one strand of a forward strand of the double-stranded DNA, and then the reverse primer will attach to the reverse strand. These will have been separated in the reaction. Then they will, the primers will then extend the strands and form a separate double-stranded DNA. And then when that happens, the nested PCR then comes and amplifies within that product. So it makes the whole reaction even more specific. I don't know if I explained it well.
Okay, another question?
Hi.
Hi.
Nice presentation.
Thank
I'm just curious as to whether the PCR assay that you use, the reference one, is that like the industrial standard, industrial benchmark? Because like I saw, you said its sensitivity is like 3%, which is I assume low. So I'm wondering, is it the industrial benchmark that you're supposed to be comparing to? And if not, what is the industrial benchmark and how does your stuff compare to the industrial benchmark?
Okay, thank you. So the PCR that we used is not the industrial standard, right? What we wanted to do was to compare our assay to an assay that targets the same region that it was designed on. So what we did was to take an assay that was designed based on the same target region that we used to see how the two would compare. Remember I mentioned that the technology is currently at TRL 6. To move it up the TRL level, we will still need to do more validations. So we will then compare the assay to the industry standard.
I'll ask a question. Is this disease region-specific within South Africa? And the samples that you used, were they across the whole country or certain areas?
Okay, thanks, Doc. When you read, literature will tell you that it's region-specific, but we obtained samples from all 9 provinces, and all 9 provinces tested positive. Yes. Thanks.
Okay, let's give it a clap. Okay, we move on to our second presenter, Rachel Brown. She is a master's student, and her presentation is generating brain organoids from mouse embryonic stem cells as a model of early neurodevelopment. Rachel, welcome.
Good morning, everyone. My name is Rachel Brown, and I'm a second-year master's student at the University of Cape Town. And this morning, I just wanted to give you a little bit of an overview of the data that I've collected during my project, which was titled Generating Brain Organoids from Mouse Embryonic Stem Cells as a Model of Early Neurodevelopment. So just a little bit of background into brain development in vivo. So brain development is a very complex process. It's quite protracted. It begins in the 3rd gestational week of development, and it continues all the way until about the mid to late 20s. What we see is the emergence of a range of different cell types from a seemingly uniform neuroepithelial cell layer. So what happens with these neuroepithelial cells is they form your radial glial cells. These radial glial cells then can undergo one of 2 processes. They either undergo gliogenesis to form your non-neuronal cell types, like your oligodendrocytes and your astrocytes, or they undergo a process called neurogenesis, which is the formation of your neurons. How this takes place is either directly through asymmetric division, otherwise indirectly through the formation of these intermediate progenitor cell types. And what you'll see throughout this course of development is an increase in the diversity of the cell types of the developing cortex. And you'll see over this time period, the areal identity of the brain is specified. So we start with a more simple structure early on in development, which then forms your more complex dynamic structure of your fully formed brain. And we see that this is a very, as I said, complex process and requires the coordination of many genetic processes. And disruptions in these processes can lead to individuals being at an increased risk of developing a neurological disease. What we see when we look in vitro is the starting point of your pluripotent stem cell line. The first structures that form are these neural progenitor rosette structures. And what these mimic are the early formation of the neural tube, where the neural tube closes to form your central canal of your central nervous system. This then further develops to form a more complex brain organoid structure, which I'll give you a little bit more information about now. And there are still some questions being asked as to how closely this mirrors our brain structure that we have once it's fully formed. But I'm going to give you a little bit more information on how we study the brain in vitro. So as you can see here, there are a range of different models that are used to study brain development, with your ideal model being your human brain tissue, either your post-mortem tissue or tissue that is resected during surgery. As you can see on the left-hand side here, we've got an increase in complexity as we move from our 2D primary neuronal cell culture models all the way to our ideal model, which is our human brain tissue. What you can also see on the right-hand side is an increase in limitation as you move from your ideal model down to your 2D neuronal cell culture model. So in the case of your human brain tissue, we see that there are some limitations associated with its low accessibility and also low throughput. It's really difficult to get access to this brain tissue, and it's also very challenging to keep this tissue alive for studies of your brain development. And again, you would only get tissue from a specific time point. You wouldn't be able to see that develop. The next more ideal model would be your brain organoids. Now, these brain organoids, I'll get into a little bit more detail about them now, but they formed from your pluripotent stem cells in culture. And you see, although they have many applications, there are still some limitations related to its limited maturation and then the organoid to organoid variability. If we look at our animal models, I'm sure a lot of you know what these entail. Obviously, there's lots of species-specific differences if you're studying human brain development using a mouse model, for example. Also, lots of ethical considerations with that. Looking at your animal and human tissue slices, the major limitation with this is the fact that it's a 2D model. Although you do have intact tissue, you don't have that same cell-to-cell and cell-to-ECM interaction that you would see in a 3D model. And then lastly, with our 2D primary neuronal cell culture, again, a 2D, that's a limitation there, and then also you don't have the surrounding supporting cell types. So you can see that there is a need for a high throughput, reliable model that we can use to study brain development. And so I'm going to give you a little bit of an overview into brain organoids, but before we do that, I'm going to give you just a brief introduction into stem cells. So there are two different types of stem cells. We either have our iPSCs, so our induced pluripotent stem cells, or our human embryonic stem cells. Now, although these two cell types have very similar properties in terms of their pluripotency, they are different. So in the case, in the case of your human-induced pluripotent stem cells, these cells are generated from your adult somatic cells, such as your skin fibroblasts. These are taken from a skin biopsy, which is then broken down and minced into your skin fibroblast cells. What happens now is you then have the overexpression of 4 transcription factors: OCT4, c-MYC, KLF4, and SOX2. And the overexpression of these 4 transcription factors allows these skin fibroblasts to revert back into a pluripotent state, namely induced pluripotent stem cells. In the case of your human embryonic stem cells, these cells are extracted from the inner cell mass of the blastocyst. So the blastocyst is the very early stage embryo, and you take these cells from the inner cell mass, you extract them and culture them. Although these models are pluripotent, which means that they can be differentiated into cells of all 3 germ layers, they do have a limitation in the fact that they are still 2D. And so this is where our 3D models of stem cells come into play. So the first 3D model of stem cells to form in culture are EBs, or embryoid bodies. And these are aggregates of your pluripotent stem cells which are formed in suspension culture. Due to the pluripotent nature of these cells, they can then be differentiated into cells of any type. Of interest in our lab are these cerebral organoids. So cerebral organoids are aggregates of neurons and glia formed in suspension culture, and they have been shown to be the ideal model to study brain development in. They have the ability to demonstrate the cell-to-cell interactions and the cell-to-ECM interactions of the brain, and also they have intact neuronal networks, which is great for your electrophysiology studies and things like that. So, how do we make these brain organoids? How this starts off is we start off with our 2D cells. In the case of this project, we use mouse embryonic stem cells. These cells are then cultured and aggregated in 96 well plates at 2,000 cells per well. The cells are then cultured in differentiation medium for the first 5 days. This differentiation medium contains ROCK inhibitor and beta-FGF for the first 3 days. ROCK inhibitor is important for promoting the survival of the cells when they're in single cell state while they're still aggregating. And then beta-FGF is also important for the formation of that initial embryoid body structure. On day 3, medium is changed and those 2 supplements are removed. And then on day 5, the aggregates are transferred into neural induction medium, which contains N2 supplement and heparin, which are both essential reagents for the differentiation of your pluripotent cells into your neural progenitor cells or your neural stem cells. We then collected the aggregates on day 11. We are still in the process of optimizing day 11 through to 18 of culture, but for the sake of this presentation, I'm just going to present data from the first 11 days. So this is what our brain organoids look like. So we start off with fairly round structures on day 1, quite light in color, and you see over the first 5 days how they start to solidify, they start to round up, and they get a lot darker in color. Then when we move our aggregates from differentiation medium into our neural induction medium on day 6, you see the formation, it's quite difficult to see on the screen, but you see the formation of these lighter buds. So what these are neuroepithelial buds. We have our neural epithelial cells, or our neural progenitor cells, that differentiate and start to migrate out, forming those initial buds, which then fold and turn to form your brain structure. So there's a bud over there, you see one up the top there, it might be easier to see, but you see the formation of these buds as these cells start to migrate out. And then you'll see on day 11, we have quite a different looking structure to what we saw on day 1. So in order to confirm what we were seeing, we wanted to do some image analysis in ImageJ. So what we did was we took images of our aggregates on every day, and we outlined them and calculated the area, perimeter, and ferrite diameter. And what you'll see here, so area, as you know, is the area of the shape, perimeter is just the border, and then your ferrite diameter is the longest point from one side of the aggregate to another. And you'll see a really nice increase in all of these different parameters. Steady increase in area and perimeter, as well as your ferrite diameter, which confirmed to us that our organoids were growing and they were developing. We then had a look at 3 other parameters, roundness, aspect ratio, and solidity. With roundness, this is just measuring how round the organoid is. You'll see a roundness of 1 is a perfectly round shape. So you'll see the organoids round up over the first 5 days, and then a decrease in that roundness as they start to bud out and form those neuroepithelial buds. The same can be said for the aspect ratio. The aspect ratio is measured differently to the roundness. So what this is you have a major and a minor axis of your organoid. These are measured perpendicular to each other. Your major obviously being your longest of the 2, and your minor being the shortest. So the aspect ratio is measured as a major over minor axis calculation. And again, you see a rounding up over the first 5 days with an aspect ratio of 1 being a perfect circle, and then the formation of these buds over the subsequent 6 days. Solidity is just showing us how solid the organoid is, and I'm sure you saw from those images, we have a really nice solid shape to begin with, and then we've got the formation of these more translucent neuroepithelial buds over the subsequent 6 days. Next, we wanted to have a look at the expression of these early neural gene markers in our organoids. So we had a look at our, neural progenitor markers, both PAX6 and nestin. We see a significant increase in both of these markers in our organoids on day 5 compared to our controls. As I mentioned previously, our embryoid bodies are spontaneously formed. They are generally more pluripotent than your brain organoids or neural embryoid bodies, and so you can see we just use those as our control for this PCR experiment. We then looked at our neuron markers, TUB3 and DCX. We see a higher expression of DCX, which is a marker of more mature neurons, which was very promising that we were seeing this 5 days into our culture period. And then what was quite interesting was the expression of REX1 in our organoids, which is actually a pluripotency marker. And I was quite surprised to see this because we are hoping that our cells are differentiating and are not pluripotent. But I suspect that this is due to also the presence of the neural stem cell population of these organoids. As you can imagine, they can't fully differentiate over 5 days, and so there is still that multipotent neural stem cell population there. Formal markers we had a look at, our midbrain and hindbrain markers. So ISL1 being your midbrain marker, OTX2 being your hindbrain marker. Again, a significant increase in expression of both of these gene markers. Astrocytes, again, significant increase in GFAP, which is a marker of our astrocytes. This is also very promising as astrocytes are important during the developmental process. Forebrain marker, FOXG1, again, significant increase in that. What's quite interesting is compared to ISL1 and OTX2, we do see a higher expression of FOXG1, which is suggesting that our organoids are taking on a more forebrain identity. So just in conclusion, our day 5 NEBs show a significant increased expression of all these neural marker genes compared to our controls. Lastly, we did some immunocytochemistry experiments. Firstly, we looked at NUN and F-actin. So NUN is it's a marker of neurons, your early neurons, and we see here a really nice expression of NUN in all of our cells. So we see this adoption of a complete neural fate. We then saw the expression of F-actin in these little luminal shapes over here. And F-actin is a marker of the cytoskeleton. So this is showing us the formation of these neural rosette-like structures that I spoke about previously, where you have this really nice little lumen forming, which is surrounded by your neural cells. We then had a look at SOX2 and TUJ1 expression, and we see, so SOX2 is a neural progenitor marker, and we see the really nice formation of these, again, these neuroepithelial buds, where our neural progenitor cells are migrating out and forming this bud, which is what we suspected from our morphology analysis. TUJ1 is a marker of both your mature and your immature neurons. You see really nice localization of TUJ1 in the outer cells. What we expected to see was these neural projections forming. However, we do know that TUJ1 is associated with nuclear cellular processes, and so that's why we see it here. We're hoping that at later stages we do see those projections forming. So just in conclusion, we saw that our MESCs can be successfully differentiated to form our brain organoids with neural rosette-like structures. This was confirmed through a range of different methods: morphological analysis, RT-qPCR, ICC. We have generated the first cerebral organoids in Africa, which is making the initial step towards creating an African-specific model to study brain development and health. And then, obviously, this work has significant implications for our laboratory and the broader African scientific community. I just want to thank my supervisor, co-supervisor, and all of our funders for their continued support. Thank you for listening.
Okay, thank you so much, Rachel, for such an interesting presentation on the brain. Okay, we'll take questions.
Okay, good morning. My name is Luando from the NGEI cluster. I wanted to actually have two questions. Being a layman in this whole topic, I wanted to understand, or wanted to ask if my understanding of organoids is correct. So you said organoids are a collection of cells that get to form the whole brain at the end of everything, right? Okay, so I wanted to understand if whether, because I've heard of the concept of 3D printing of human organs, and that it stems from having to donate like cells from, if let's say the organ that is to be developed is for you, so they will need to take cells from your existing organ and then they can replicate that. So that does this study of brain organoids also correlate with the idea towards 3D printing of human organs? Can that be something that can be achievable with what you are doing here?
Yes and no. So what we are doing is we are allowing these organoids to spontaneously differentiate. Obviously, we are directing them towards a neural ectodermal lineage, which means that your mesodermal cell types and things like that will not be present in the organoid. So it provides kind of room to co-culture and produce an organoid that then has all of those cell types. With your 3D bioprinting of organoids, that obviously is a little bit more controlled. You've got a very specific amount of cells that are being added of a specific cell type. Obviously, you do need to make sure that you are using cell types from the same individual, that you don't have any of that interaction happening. So in the sense, yes, that is a different, it's still an organoid, but it is a different stream, because here we're working with like spontaneous generation, because we're wanting to see if we provide it with what it needs, what's going to spontaneously happen, and that will inform what happens during early neural development, which we can't obviously see in vivo, which could then inform, okay, at this point, this cell type, so this is when our astrocytes come in, which will then inform how we do the 3D bioprinting. Does that make sense? Okay.
Thank you for a very nice talk, very interesting. I'm interested in finding out, or rather curious, the endpoint is, of course, to use these as a model for
Sorry,
Does-
I missed that.
the endpoint is to use these as a model?
Yes.
Yes. So how long are the organoids viable? So if you were to create your organoids and then want to test them, or test a treatment or whatever your end goal is, how long do they last?
So generally, in the global north, they've managed to culture these organoids for 300 days, so for a very extended period of time. Obviously, that's very resource intensive and requires a lot of work. So generally, while they're still in culture, they are viable. As long as you're feeding them, as long as you're giving them what they need, they do generally stay viable. But then if you're wanting to do other downstream, so like for example ICC, we obviously fix them, but if you're wanting to do your electrophysiology tests and stuff, you need to make sure that you keep them, because they can die quite quickly when you remove them from their culture medium. So you would have to figure out a way, I haven't done that personally, but you'd have to figure out a way to make sure that they stay within their culture conditions. Yeah.
One last question.
Good morning, Rachel.
Morning.
Great presentation. I just have one question. So what are the biggest limitations of using mouse embryonic stem cells derived organoids in comparison to human organoids?
So obviously, you've got the major difference, which is the species-specific differences. Our lab, we wrote a review which compared the timing between your human organoids and your brain organoids. And generally what happens is your mouse organoids tend to develop a lot faster than your human organoids. So in terms of that timing, you're going to have it happening a lot quicker. So that is a limitation. Ideally, we are moving in the direction of using human cells to create human organoids, because ultimately that is more appropriate to study human development. But for now, what we're seeing are promising results that's mimicking what happens in the early development of the mouse brain. Yeah.
Okay, let's give our speaker a round of applause.
Okay, the last speaker of the session is Potsiso Koma, and her topic is investigating the antioxidant, mycobacterial and antibiofilm activities of medicinal plants against Mycobacterium smegmatis.
Good morning, everyone. My name is Pujiso, a PhD candidate from the University of Limpopo, and today I'll be taking you through my presentation titled Investigating the Antioxidant, Antimycobacterial and Antibiofilm Activities of Medicinal Plants Against Mycobacterium smegmatis. Tuberculosis is an infectious airborne disease caused by Mycobacterium tuberculosis complex, which primarily affect the lungs. It is resistant to the current available treatment methods, and this is due to it developing new survival mechanisms, bacterial cell-to-cell communication, which promotes biofilm formation and dispersion. The biofilms act as a physical barrier, preventing antibiotic penetration and protecting the bacteria from environmental stressors. The first-line drugs induce side effects, which makes it difficult for patients to continue with the treatment for a minimum period of 6 months. So this led to research to uncover new alternative methods to treat and manage TB using medicinal plants. Traditional medicinal plants have been reported to have bioactive compounds that can be used as alternative sources of therapeutic agents to control and manage TB. The study aimed to evaluate the antioxidant, antimycobacterial, and antibiofilm activities of medicinal plants against Mycobacterium smegmatis. Here are my objectives. The plants were collected at the Lowveld National Botanical Garden in Mpumalanga, and some of the plants were collected within the University of Limpopo. They were then air-dried at room temperature, ground to fine powder, and 1 gram of the plant leaf material was extracted with solvents of varying polarities. They were then transferred into pre-weighed vials, and the mass of the extracts were calculated. And Figure 1 shows the mass extracted by the different solvents, and it was observed that methanol had high extraction yield as compared to all the other extracts, except for seed labrum and Eucubectia capensis, which had their high extraction yield from the water extract. This shows that these plants had more polar compounds because the methanol and water are polar solvents which tend to extract more polar compounds. The extracts were then reconstituted to 10 milligram per mil with acetone, and phytochemical analysis, quantification of phytoconstituents was done using the standard chemical methods. It was observed that Dicrostachys cinerea had the highest total phenolic and tannin content, Hypericum ruperianum with the highest total proanthocyanin content, Buddlegia salvifolia with the highest flavonoid content, while Combretum apiculatum had the highest flavonoid content. Since these phytoconstituents have been reported to have antioxidant, antimicrobacterial, and antibiofilm activities, we then looked at the antioxidant activity using the DPPH free radical scavenging activity assay, whereby in this assay, plants with antioxidants transfers a hydrogen atom to the hydrazine, and the nitrogen atom of DPPH is reduced. It forms a purple color when dissolved in methanol, and the presence of antioxidant compounds are shown by the yellow bands against the purple background. We then have to quantify the amount of oxidant that will be able to inhibit the free radicals of the reactive oxygen species. And it was observed that Dicrostachys cinerea had activity comparable to that one of ascorbic acid, which then shows that the antioxidant activity from Dicrostachys cinerea might be from the total phenolics that were quantified in the plant. With the ferric reducing power assay, the plants with antioxidants reduces the ferric ions to ferrous ions, and it forms a brisant blue color. Ascorbic acid had the lowest IC50 value, followed by seed labrum. We then looked at the antimicrobacterial activity using the broth microdilution assay and bioautography assay, whereby INT was used as an indicator. It is based on the dehydrogenase activity of viable cells to convert the INT into a formazan color. The clear bands against the pinkish background indicates where the phytoconstituents within the plant extract were able to inhibit the growth of Mycobacterium smegmatis. And it was observed that Buddlegia salvifolia, Carissa macrocarpa, Compritum apiculatum, and Buddlegia saligna had promising bands as compared to other plants. We then looked at the minimum inhibitory concentration and minimum bactericidal concentration, whereby the minimum inhibitory concentration is the lowest concentration of the antimicrobial agent that will be able to inhibit the growth of the bacteria. And with the minimum bactericidal concentration, it tells us about the minimum concentration that is needed to kill the bacteria. It was observed that Combritum apiculatum had the lowest minimum inhibitory concentration of 0.16 milligram per mil, which was similar to the minimum inhibitory concentration of the rifampicin. Ooh. Of the rifampicin. And it also had the minimum bactericidal concentration from the same extracts of 0.63 milligram per ml. We then looked at the combination effects to check if whether the positive control and the plant extract can work together to inhibit the growth of Mycobacterium smegmatis. It was observed that a combination between Hapaphilum cafram and rifampicin, Combretum apiculatum and rifampicin had an additive outcome. A combination between Dicrostachys cinera and rifampicin had an antagonistic outcome, while the other combinations had an indifferent outcome. Looking at the antimycobacterial activity from bioautography and broth microdilution assay, 5 plants were chosen for further analysis, which are Badlegia saligna, Hapaphilum cafram, Badlegia salvifolia, Combretum apiculatum, and Carissa macrocarpum. The effect on growth phases and inhibition of growth was evaluated using the growth curves at 2 times MIC, MIC, half MIC, and quarter MIC value. It was observed that all the plant extract had good activity against Mycobacterium smegmatis. Since this method does not tell us about the number of viable cells within the extract, we then looked at the enumeration of viable cells using the time kill assay. Only one plant, the positive control and the untreated, are shown on the slide. And it was observed that on figure 5A, there was countable number of colonies from time of inoculation until 18 hours, as shown in the graph. And then at 24 hours, there were too many to count colonies. With figure 5B, which was the positive control, the rifampicin had a bacteriostatic effect against the organism for a period of 9 hours. And then after 9 hours, at 12 hours, there were countable colonies until 18 hours. And with the untreated, which is figure 5C, there were too many to count colonies. Mycobacterium is associated with exopolysaccharides, which plays a huge role in motility and biofilm formation. The quantification of exopolysaccharides was done using the phenol sulfuric acid assay. Where in this assay, carbohydrates react with sulfuric acid to produce monosaccharides, which then reacts with phenol to produce a colored complex, and the color intensity is directly proportional to the number of exopolysaccharides present. It was observed that less exopolysaccharides were quantified at MIC value as compared to half MIC value. And Badlegia salvifolia had the lowest number of exopolysaccharides quantified. In motility, these exopolysaccharides Mycobacteria uses a sliding motility to move around. So in motility, these exopolysaccharides play a huge role to prevent the bacteria from friction by the surface area. So it was observed that there was 100% inhibition at MIC and half MIC value when it was treated, when Mycobacterium smegmatis was treated with rifampicin, followed by Hapaphilum cafram with 82% and 71% at half MIC value, while Combretum apiculatum had the lowest inhibition at half MIC value. In biofilm formation, these exopolysaccharides play a huge role as they contribute to the formation of an extracellular matrix which shield the bacteria from the antibiotics. The prevention of biofilm formation was evaluated using the crystal violet staining assay. And it was observed that there was no significant difference between the ability of rifampicin and Combretum apiculatum to inhibit initial cell attachment at all the concentrations. Low activity and enhancement of activity. Enhancement is the one with the negative inhibition, was observed when the plant extracts were treated with Hapaphyllum cafram, Badlegia salvifolia, Carissa macrocarpa, and Badlegia salignum. Looking at the prevention of biofilm formation, it was observed that all the plant extract had good activity with an inhibition greater than 50%, except for Badlegia salvifolia at MIC over 8. We then looked at the eradication of the mature biofilm. And it was observed that there was no significant difference between, the Combretum apiculatum and the orange one, and Badlegia salvifolia had good inhibition with an inhibition percentage greater than 50%. Low activity and enhancement of activity was observed from the other extracts. In conclusion, the selected medicinal plants exhibit potent antioxidant, antimicrobacterial, and antibiofilm activities, indicating their potential as sources for isolation and characterization of bioactive compounds. Here are my acknowledgements. Thank you.
We'll take questions. The first question.
Thank you for interesting talk. I have to confess that most of it went over my head. So maybe just reducing it to standard grade for me. Please clarify, so in the pictures that you showed where you were showing the bacterial growth, your plates. Yes, so were you saying that maybe the media, was it treated with now your plant extract, and then the bacteria only grew, the growth of bacteria was delayed?
Yes.
So does that mean that in terms of now treatment, that also dictates like how often the patient will have to take the treatment?
Yes.
Okay, thank you.
Hi. Out of interest, I would like to ask, so you obtained secondary metabolites from your plant extract, right?
Yes.
So I wanted to ask if these secondary metabolites were already existing in your plant extracts, or they were existing from possible prior exposure to the M. smegmatis strain before. So I don't know if I'm making sense.
You mean these ones, right? No, they were existing in the plants.
So it's not from exposure whatsoever?
Exposure might be from the sun, environmental conditions.
Thank you so much for the presentation. I just want to check what informed your choice of plants.
The plants were selected based on literature about the ethnopharmacological use to treat respiratory infections and their related symptoms. Then we looked at the plants that are less investigated, and we chose those ones.
Just a follow-up question from that one. Is there a particular decision that made you choose specifically the leaves other than maybe the stems or the roots?
Based on availability. Because if you take roots, the Environmental Act will not allow you. So you only get less amount. And also in this study, I'll still have to isolate the bioactive compounds. So if I have to use the roots, it means it will be something else. We won't have the plants at some point, yeah.
Hi. Did you purify your extracts out of the acetone?
Did I do it?
Did you purify the acetone out of your extracts?
Come again?
The acetone, you extracted them with acetone? The extracts, you extracted them with acetone?
Yes.
Did you purify the acetone out?
No, because we extracted with the acetone. And then from there, we let the extracts dry so that we can get the mass. Then we reconstitute with acetone because of its feasibility with polar and non-polar compounds. So it allows us to extract more compounds.
You reconstituted with acetone and then treated, or rather plated that?
Yes.
So how can you be sure your bacteria didn't grow because of the extracts rather than the acetone?
Oh, sorry. We have acetone as the negative control.
Okay, cool. Did you do any toxicity testing on the compounds?
I'm still busy with that.
Okay.
Morning. Thanks for a great presentation. You mentioned that if you were to use the roots, you'd have to isolate, right? So, meaning in this case, you didn't isolate, you just took the extract as it is, and then you are working with the whole...
The crude extracts, yes, for now. But I'm busy isolating.
Oh, okay. So meaning that you're going to look at which of your bioactives is more active?
Yes.
Oh, okay. Thanks.
Just last one. Just to check. Have you published any of this data yet?
Not yet.
There's no publication yet on this stuff. Okay.
Thank you so much for this.
Let's give our presenters one more round of applause. Yes, our session ends here. Where's our... Okay, next we are proceeding to the poster and exhibition. So we can go and view the posters, ask questions, communicate with the poster presenters. Thank you once again.